Hello everyone, my name is Taklahana. It's my absolute pleasure to be presenting this journal by title of Cell-Free Metabolic Engineering Biomanufacturing Beyond the Cell. First, I will start with a quote by the Department of Chemical and Biological Engineering in Northwestern University in Illinois. They said that today's chemicals and materials are almost entirely derived from fossil fuels and their products. They used the petrochemical industry as an example of how there is a high demand and supply limitation. There is really no way that we can control the product of petroleum. Once it's done, it's done. We can't get more because it's one of the generated resources. Similarly, in the industrial biotechnology and microbial field, there is a high demand for low-cost chemicals and natural products, but there are some major consequences. First, low volumetric productivities due to the cell capacity of production. Second, the buildup of toxic intermediates. Third, byproducts loses via competing pathways. Just looking at the three limitations above, the problem is the cell. So the solution is to find or create an alternative to the traditional bioengineering model using vitro assemblies. The cell-free metabolic engineering or CFME system has created a way in which the product can be created without using the cell or in vitro. First, they express the enzymes needed. In other words, they make copies of the cell that contains the enzymes needed for the process. Second, they take half of the copied cells and they go through purification, where they extract enzymes that needed for single reaction and recombine them to construct a certain pathway. The other half will go through lysate preparation to obtain the crude extract, where it contains all the other components needed for the pathway, like cofactors and proteins, etc. Third, the purification and the crude are mixed in a way that it will optimize the production in regards of the amount and speed. This way, we have created a pathway that does not rely on the cell in process of generating the product as it's shown in the image on the right. The importance of this work lies in the three limitation keys that I have mentioned in the beginning. Using the CFME system, we have increased the yield by controlling the ratios of the crude extract and the purification product. We have avoided the toxicity constraints and competing pathways by choosing only the important proteins, cofactors, and enzymes during the lysate and purification at the purification step. Moreover, we now have more control over the product, which provides on-demand use of manufacturing. As fascinating as this work sounds, it's still having pros and cons. First, it's still unclear to what extent can the CFME system work commercially, because it's new and has not been tried commercially yet. Second, the protein purification process is very expensive. Third, the stability and longevity of reaction cofactor regeneration is still undetermined. On the other hand, there is no viability constraints, the reaction conditions are controlled, and it's easy to add the substrate and easy to remove the product. At the end, I hope you guys have enjoyed the presentation as much as I have enjoyed the paper. Thank you.